Good afternoon, everyone. So I am Seemadri, and as Julia rightly said, I'm the program doctoral fellow at Fields for the thematic program on mathematical oncology under the supervision of Professor Shivlokanathan. Uh, but today I will be talking about a project which I did in my PhD back home at the Indian Institute of Science with Professor Mohit Kumar Jolly, and the experimental validation of the work was done at Duke University in the lab of Professor Jason Somarelli. So this would touch upon some of the hallmarks that Professor Thomas talked us in the first day itself, such as phenotypic heterogeneity. I would also talk about metastasis, invasion, drug resistance, and also immune evasion. And I'll try to connect various hallmarks together to provide some sort of mechanistic understanding. A disclaimer, sorry, before that, so my ex-supervisor would often say that identity crisis is his identity. So in the past few days, when I introduce myself and tell my background, I'm sort of stepping on the identity crisis platform where I don't belong exactly in one category, but I have experience in several bins. And so cutaneous malignant melanoma that arises from a fully differentiated pigment-producing melanocytes is a deadly and highly aggressive form of skin cancer, quite infamous for its high metastatic potential. And melanoma samples, they show intra and intertumoral heterogeneity, which means they comprise of diverse phenotypic subpopulations, differing in their functional profiles and molecular attributes. Say they have differing proliferation rates, differing metastatic potential, different immune susceptibility, and all of these have their underlying genomic and non-genomic basis. But for the purpose of this study, we have specifically focused on the non-genetic phenotypic aspect of melanoma heterogeneity that arises as a result of one phenotype transitioning into the other, in this case, proliferative to invasive transition. So melanoma samples, they can reversibly switch from a proliferative cell type to an invasive cell type, aiding in drug tolerance, immune evasion in patients with progressive disease. However, recent characterization of melanoma cell lines and tumor samples at both single and bulk resolution showed us that, the phenotypic heterogeneity in melanoma, it extends beyond just proliferative or just invasive, and cells can dynamically acquire a spectrum of phenotypes along the proliferative invasive axis. Say we have, till now we have 4 subtypes that are determined. There's one paper which talks about another, but nothing much substantial after that paper. So I'll just focus on these 4 subtypes: melanocytic and intermediate in the proliferative category, in neural crest and undifferentiated, which are broadly invasive. So the first problem statement from this was, how do the phenotypically plastic states arise in melanoma? Now, to answer this, we took a bottom-up approach where we modeled a gene regulatory network that comprises of transcription factors that are commonly implicated in this proliferative to invasive transition. And then we incorporated the literature-derived regulatory links among them. For example, MITF inhibits ZEB1, so all the hammerheads are basically repression and all the arrows are activation. So now to understand how this gene regulatory network behaves, how do genes interact and influence each other, which genes will be co-expressed together, which will not. We used a simulation framework which is called a RACP, as Harvey likes to call it, which is random circuit perturbation. It was developed in Herbert Levin's lab at Northeastern University. So what RACP does is basically takes the topology of the gene regulatory network. What I mean by topology is the number of genes and the regulatory links among them. And then it models it as a set of chemical rate-based nonlinear ODEs, which have a production. Let's suppose A inhibits B, B inhibits A, which is a typical toggle switch. So it is basically modeled as a production term. The inhibitions are modeled by a Hill function, and then you have the degradation term. And you write such equations for the entire gene regulatory network. You solve them, and then you end up with steady-state expression patterns. So once you obtain the ensemble of steady-state expression patterns, it is basically a possible phenotypic space, all the phenotypes that can be enabled by this gene regulatory network. Now, I can represent all of my steady states, or as I like to call it, each row can be construed as a virtual cell. So in a heat map, you can see that some of the genes are co-expressed together, some are not. Like if you do a hierarchical clustering on the heat map, you can see that MITF and SOX10 are co-expressed together, and SOX9, June, and ZEB1 are co-expressed together. So each row is basically one steady state expression pattern. So you have different initial conditions, different parameter regimes, and these are like all 10,000 solutions. And sorry?
Solutions of what?
So each solution is basically the level of each transcription factors. So here you can see that, so if we if we concentrate on the first row, the MITF expression is high, the SOX10 expression is high, but the other three are low expressions. So,
sorry for the questions.
Yes.
And personally, we can actually ask questions.
Yes, sure.
Well, you have a 5-dimensional system.
Yes.
It cannot have 10,000 steady states.
So you initialize in different, you have initial conditions, like you start with a particular level of each gene, and then you look at, and then you have those Hill coefficients and, fold change, you have different parameter regimes that you sample from, and then you end up in. So qualitatively, we can see there are 4 states. For example, in this set of solutions, you see that MITF and SOX10 are always high, whereas the other 3 are low. In, for example, this sort of solutions or ensemble of solutions, you see that MITF and SOX10 are low. However, the other 3 are high. So in order to better classify these solutions, or as I would like to call it, virtual cells, into phenotypes, I devised 2 scores, which are basically proliferative score, which is the Z-normalized steady-state expression of these 2 transcription factors or proliferative master regulators, and invasive score, which is basically the summation of Z-normalized steady-state expression of the other 3 genes. And then you project all of your virtual cells in this proliferative invasive plane, and you can see that you have sort of qualitatively 4.
Number -6 mean, for example?
Sorry?
I want to understand the units. What's
So units are, because it has been Z-normalized, so -6 means it's very low on the invasive score. So if you have like a cell here, it is it's a low phenotype. It's low proliferative, it's low invasive. So basically, if you look at cluster of cells here, they are high, they have high proliferative score, but they have low invasive score. So it is
trying to understand the scale.
Yeah, the scales like all the expressions we have taken, and then we have just normalized it. So
Normalize.
renormalization across all the cells.
What does that mean? Sorry.
So you take the concentrate, like whatever concentration of genes you get at the end, like the steady state solutions, you just normalize the expression across the, so you have 10,000 cells. You just look at all the 10,000 cell expression and you just standardize it or no.
Sorry, following up on
Yes.
that. So from my understanding, if you're 0, 0, you're basically average. You're average.
Like sort of, both invasive and both proliferative. So you would get essentially your intermediate phenotypes there. So your neural crest and intermediate. So these are your like intermediate phenotypes. Yeah, so right now, at this point from our gene regulatory network, we are sort of able to deduce the 4 phenotypes, but does it actually correspond to what we see in data? So every time we model something, we validate with data. For that, we do transcriptomics analysis at both single and bulk resolution. So this is basically 10 melanoma cell lines, which have been derived from melanoma patient cell cultures. And if you look at in all of these cell lines, if you look at the gene expression, so these are like real cells. This is computational. These are virtual cells, but right now I'm talking about real cells. So if you look at the gene expression of all of these 5 transcription factors, you see that there's bimodality. So you can basically binarize their expression as 1 or 0. Because you see there's if you just plot their expression, you see bimodality. So what I did is I sort of binarized each of these expression of genes in the cell lines. So if you look at the red cell line, in this cell line, you would find that MITF levels were higher, SOX10 level was higher, and the other three gene level was lower. So that is what we see in our simulation also. It's a classical proliferative phenotype. However, if you look at, the MM099 cell line, this has you would find that the proliferative master regulators are low and the invasive master regulators are high. So this is classically invasive phenotype, which you also see in the model. So there's this antagonism between the proliferative and invasive axis, which we were able to see in our model and were able to validate in single cell resolution as well. I can talk more about it, but because I have to cover a lot of things, maybe I'll just skip to the next part. Yeah, so even before going to single cell resolution, we look at bulk data. So CCLE is basically all the cancer cell line, and TCGA is basically all patient tumors. And you would still see that even if you define the proliferative score with just 2 genes or invasive score with just taking 3 genes into consideration, you can see that there is this good antagonism that you see. But we are not the first groups who are trying to define these phenotypes. Long back, there are 2 seminal papers, which is by Verfeli and Hoek, and they the way you define phenotypes in bioinformatics is you look at the differential expression of genes, and then you define a gene signature, and then you look at the scoring of that gene signature. So initially, Verfeli and Hoek both have defined their gene signatures, which are like 600 plus genes. So if these genes are sort of upregulated, or you would say you get a higher scoring, then that is like if the proliferative signature is higher, you would anticipate that, okay, that's a proliferative phenotype. But if you actually want to test in lab, then no lab will allow you to test 600 genes. So what is really nice about this exploration is if you correlate our proliferative score, which is just 2 genes, with the Verfeli proliferative score, which has like 600 plus genes, you actually get a good correlation. And that is both in cell line as well as tumor samples. So the 5 genes that we have a very minimal gene regulatory network that was sufficient to capture the major patterns of proliferative invasive nature that you see in melanoma samples at cell line and patient tumor levels. So now that we were roughly able to, model the different phenotypes, we wanted to understand what happens if you perturb this gene regulatory network. The way it is done in clinics is, they can downregulate some gene, they can put a siRNA, and they can, inhibit that gene, or they can upregulate and overexpress certain gene. So we try to do that in simulations. The way we do that is we basically change the fold change in the Hill's regulatory term. So in the first case, what we did is we down expressed SOX10, and we then compared it with our wild type network. So the moment you down express SOX10, you are seeing that the proliferative population decreases, the invasive population increases, the neural crest decrease, and intermediate population they increase. And do we actually see that in data? So we also analyzed a transcriptomics profile where they have done SOX10 downregulation, and we see that the similar trends are being shown. Next, we did is we overexpressed SOX9, and we saw a reversal. We saw a reversal where you see invasive population going up, and we also see that in data. Now we wanted to see whether transition is happening or not. For that, we need to study a time course data, for which we studied in this case where they have knocked down SOX10 and they have collected data at 24 hours interval. So you have a control data. So the wild type cells which did not have SOX10 knockdown, they are much more proliferative to begin with. As you take the reading after SOX10 knockdown, they are progressively transitioning to a transcriptional state that is more invasive. So that basically shows that the switching can also happen. Now that we have looked into one of the hallmarks, which is, phenotypic heterogeneity, we thought of connecting phenotypic heterogeneity with the immune suppressive capabilities, because that's also one of the hallmarks, and we're trying to understand that. So all of these phenotypes have been known in literature to have different levels of immune suppressive capabilities. And we wanted a mechanistic or mechanistic understanding for it, for which we looked at PDL1, because PDL1, or programmed death ligand 1, is basically an immunosuppressor protein which is present on the surface of your cancer cells. Now, PD1 is another receptor, which is a receptor protein which is present on the surface of T cells. The moment PDL1 interacts with PD1, it leads to T cell exhaustion. So you have all these immunotherapies such as anti-PDL1 or anti-PD1, which goes and binds to one of the receptors, so that the interaction between PDL1 and PD1 is sort of stopped. So for that, we integrated PDL1 and interferon gamma into our gene regulatory network, because we know that interferon gamma sort of transcriptionally activates PDL1 and vice versa. And then we again simulated this gene regulatory network using the previous framework. And what we were able to see is, in the proliferative phenotype, PDL1 levels are lower. However, in the neural crest and invasive phenotype, the PDL1 levels were higher than the proliferative. It was sort of mid to high range of PDL1. And that is something we also see in data. So we see that as the cells become more invasive, their PDL1 levels sort of increase. And there's also variability in that. So more invasive, sort of more PDL1. Next, we wanted to see if our model could, capture the positive association between interferon gamma and PDL1. And we see that in basically the neural crest and undifferentiated or invasive cells, you have higher levels of interferon gamma. So more invasive, more PDL1, as well as more interferon gamma, more PDL1. Now now we wanted to segregate these two axes, and we sort of devised two situations. One where you have a low interferon gamma regime, and one where you have a high interferon gamma regime. Now, in the low interferon gamma regime, we look at cases. I still have time, right? Like 5 minutes, okay. In the low interferon gamma regime, you look at the PDL1 levels in all of those 4 phenotypes. You see that in your proliferative and intermediate, PDL1 level is lower, but in your neural crest and invasive, your PDL1 is like mid to high. But the moment you switch on to a high interferon gamma regime, even your basal PDL1 level increases, even in proliferative and intermediate, but the increase being higher in more invasive sort of phenotypes. So you have 2 axes. One is your interferon gamma axis, and one is your phenotypic heterogeneity axis, which basically controls PDL1. And we found that there's a combinatorial sort of control, where if you look at, this is basically patient data, and if you look at the blue cluster, which is, I'm sorry, I'm rushing, but I can always, catch up after this if you want to understand more. If you look at the blue cluster, which is high phenotype, it is more invasive, and it has high interferon gamma, that basically has the highest level of PDL1. And if you look at the pink cluster, which is a low cluster, which is more proliferative and has less interferon gamma, that has the lowest amount of PDL1. That is what we derived from model, and then we were able to experimentally validate it with the help of Jason, where we treated each of the cells with TGF-beta and TNF-alpha to induce this phenotypic transition, and interferon gamma as an independent modulator of PDL1. And you see, wherever there is this combinatorial treatment of TGF-beta, TNF-alpha, which pushes the cells to a more invasive phenotype, and interferon gamma, the PDL1 levels basically upshoots up, and we can see a synergistic control. Maybe I will skip this part of cell resistance because I don't have time, but happy to chat about it.